Welcome to this Hand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive into today's fascinating topic, please take just a moment to like, subscribe, and maybe share this deep dive with friends and family. It really helps support the Hand Science Experience, and let's just keep bringing you breakdowns of complex science.
So today we're tackling a huge question, human aging, but we're coming at it from a slightly different angle, not just genetics, not just lifestyle.
Right, we're looking deeper at the functional level through the lens of the proteome.
Exactly, the proteome, all the proteins actually doing the work in our cells.
Yeah, if you think about it, DNA is the blueprint. RNA is like the instructions copied from that blueprint.
Yep.
But the proteins, they're the workers, the machines, the actual structures. They're what gets things done. So we're looking at the real output.
And our guide for this is a really foundational study. It was published in Cell by Ding and colleagues just this year, 2025. They didn't just look at one or two things. They aimed to create a massive proteomic and histological atlas of aging.
And massive is the word. Get this. They mapped over 12,700 distinct proteins across more than 500 samples covering 13 different tissues. We're talking cardiovascular, digestive, immune, skin, muscle, you name it.
And critically, across a huge age range, 5 decades, from age 14 all the way up to 68.
Wow. So the core question they were asking was, can tracking these proteins give us a real systems-level map of how we age and maybe even where it starts?
That's the goal. And, the findings, they're pretty illuminating. We're going to talk about evidence for kind of aging inflection point around middle age.
Okay.
And this really interesting idea of a primary Senohub, like an aging command center in the body.
A Senohub.
Yeah.
Intriguing. All right, let's start with maybe the first big surprise they found.
Yeah.
About the blueprints versus the reality.
Right. For a long time, a lot of research focused on the transcriptome, the RNA messages. The assumption was, well, that tells you what proteins are being made.
But it's not always that simple in biology, is it? What did the protein data show about that link as people got older?
It showed this age-dependent decoupling. So the overall match, the correlation between mRNA levels and the actual protein levels, it was only okay to begin with. Maybe 0.4, 0.5 correlation.
Moderate.
Moderate, exactly. But the key thing was this correlation got weaker as people aged, especially in certain tissues like the spleen, muscle, lymph nodes.
So the older you get, the less reliably the RNA instructions predict the actual protein landscape. The factory is getting sloppy.
That's a great way to put it. The quality control seems to falter. And that leads directly to this concept of declining proteostasis.
Proteostasis, the cell's ability to maintain healthy proteins.
Precisely. Keeping proteins folded correctly, clearing out damaged ones. They saw a clear drop in the machinery responsible for this.
Like what specifically? What parts of the protein production line were breaking down?
Well, they saw fewer proteins involved in making new proteins, things like ribosomal proteins, both in the cytoplasm and mitochondria. So fewer assembly workers, and maybe even more critically, fewer proteins involved in folding and transport after they're made. Things like chaperonins, CCT5 and CCT7 were mentioned specifically.
Chaperones, those are the molecules that help proteins fold into the right shape.
Exactly. They're crucial for quality control.
Okay, so if the assembly line and the quality control are failing, you're going to get buildup, molecular garbage.
Worse than garbage, really. Toxic buildup. They observed accumulation of misfolded proteins, particularly amyloid structures. SAA1 and SAA2 were highlighted.
Amyloids, that rings the alarm bell. That's linked to all sorts of age-related diseases.
It is. And when these amyloids accumulate, it seems to trigger a massive and kind of inappropriate immune response. Lots of immunoglobulins, lots of complement proteins like C4A.
So the mess isn't just sitting there, it's actively inflaming the tissue, setting off alarms everywhere.
That's the picture they paint. They actually proposed this idea of an amyloid immunoglobulin complement axis.
An axis, so a linked system.
Yeah, a kind of vicious cycle. Bad protein quality control leads to amyloid buildup, which triggers the immune system locally, causing inflammation, which probably makes protein quality control even worse.
Okay, that makes sense at the tissue level. But did they see any patterns across different tissues? Like universal signs of aging popping up everywhere?
They did, and this is really important for finding biomarkers. They found 2 main groups of proteins changing consistently with age, pretty much regardless of the tissue.
All right, what was going down everywhere?
Those they called UDPAs, ubiquitous downregulated proteins with aging. About 31 proteins that consistently decreased in 6 or more tissues.
Any key examples?
One they mentioned was PRPF4. It's involved in pre-mRNA splicing, which is like a super fundamental step in making almost any protein. So a core process seems to erode.
Okay, and the flip side.
Yeah.
What was going up everywhere? Let me guess. Inflammation, damage response stuff.
You got it. The UPAs, ubiquitous upregulated proteins with aging, they found 29 of those. And yeah, heavily linked to inflammation, extracellular matrix changes, that kind of thing. A standout example was serum amyloid P component, or SAP. It's actually part of those amyloid deposits and helps activate the complement system.
So declining core functions, increasing inflammation, and damage markers. It sounds like the body's trying to patch things up, but the patching process itself causes problems.
That really captures the paradox of aging, doesn't it? Protective responses becoming harmful over time. But the cool thing is, having these universal markers allowed them to do something predictive. They built proteomic age clocks.
Ah, okay. Using these protein signatures to estimate biological age, not just chronological age.
Yeah.
How well did those work?
Remarkably well. Based on the protein panels, they could predict a person's chronological age with really high accuracy. Spearman coefficients mostly between 0.74 and 0.95. That's pretty tight for biological data.
Very tight. It means these protein signatures are a strong indicator of how old a tissue really is, biologically speaking. Did any single protein stand out as being important for these clocks across many different tissues?
Yes, one really popped out, TMP3, tissue inhibitor of metalloproteinase 3. It was a key predictive feature in the clocks for 9 different organs.
TMP3. Okay, follow that one away. Seems like a potentially powerful universal aging marker.
Definitely one to watch.
All right, let's shift from the what to the when. Did they see these changes happening gradually, or was there a specific time frame?
This was maybe one of the most striking findings. When they looked at the timing, the temporal analysis, it wasn't just a slow, steady decline.
No.
The data suggested that many tissues undergo a really substantial, almost synchronized wave of proteomic change right around age 50.
Age 50.
Specifically.
Well, the window they identified was primarily between 45 and 55 years old. But yeah, a major transition point seems to happen around then.
Wow. That puts a number on something a lot of people feel, doesn't it? That middle age isn't just a number, it's a biological shift. It's not a gentle slope, it's more like a steeper drop-off. The data certainly points that way. A point where the body's systems, particularly proteostasis, might cross a certain threshold, accelerating some of these aging processes.
That's huge. It gives a potential window for intervention, doesn't it, before things really start to snowball.
Absolutely. Targeting that transition phase could be key.
But we know aging isn't uniform. Different parts of the body age differently. Which brings us to this Senohub idea you mentioned.
Sure.
Did one tissue stand out as aging faster or earlier?
Yes. One tissue really showed the most dramatic and continuous changes across the whole lifespan studied.
And that was?
The aorta, the main artery coming out of the heart.
The aorta. Not the brain or muscle or immune system. The aorta.
The aorta. Out of all 13 tissues, the cardiovascular system, specifically the aorta, showed the biggest proteomic shifts over time. It seemed particularly susceptible to aging and seemed to age early.
That's counterintuitive almost. We often think of arteries as being damaged by aging, by high blood pressure or cholesterol, not as a source of aging.
Right, but that's what led them to propose this Senohub concept. The idea that maybe certain tissues, like the major arteries, are highly sensitive to aging triggers, age faster, and then they start influencing the rest of the body.
How would they do that? How does an aging aorta affect, say, your muscles or spleen?
By releasing signaling molecules into the blood, senokines, basically chemical messages that spread the aging signals systemically.
Okay, so they must have looked for specific signals coming from the aorta.
They did, and they found a prime candidate, a protein called GAS6, growth arrest specific 6.
GAS6. Okay, what's the evidence linking GAS6 to the aorta and systemic aging?
Well, first, they found GAS6 levels were high in the aging aorta tissue itself, and second, importantly, GAS6 levels were also elevated in the blood plasma of older individuals.
Suggesting the aging aorta is pumping it out into circulation.
That's the strong implication. It's the messenger carrying the aging news from the aorta to everywhere else.
But correlation isn't causation. Did they test if GA6 itself could actually drive aging phenotypes?
Yes, this is the crucial experiment. They took middle-aged mice and injected them with recombinant GAS6 protein.
And what happened?
The effects were pretty clear and quite rapid. The mice showed impaired physical function, weaker grip strength, worse coordination.
Just from this one protein that's originated from the vascular system.
Just from GAS6. And it wasn't just physical performance. They also saw molecular signs of accelerated aging in multiple organs. More DNA damage markers in the aorta itself, yes, but also structural problems in the spleen and liver.
Wow. Okay, that really strengthens the case for the aorta acting as a xenohub, using GA6 as one of its main tools to broadcast aging signals.
Exactly. It connects the dots from local tissue decline, the proteostasis failure in the aorta, to systemic problems via these circulating factors.
And was GA6 the only one? Or were there other potential messengers?
GAS6 was a major focus, but they did identify other proteins accumulating in the blood with age that seem to promote senescence, especially in blood vessel linings. Things like GPNMB, COMP, HTRA1, and nano-TCH3 were also mentioned. These are sometimes called humoral senoproteins.
Humoral, meaning circulating the body fluids, like blood.
Correct.
So it's really painting a picture of aging as this interconnected network problem. The aorta gets old first, maybe triggered by proteostasis issues, and then it starts sending out these inflammatory signals like GAS6, which then accelerate aging elsewhere.
That seems to be the emerging picture, yes. A cascade effect, with the vascular system potentially playing a really central driving role.
So let's try to pull this all together for everyone listening. We started with this huge protein map across tissues and ages.
Right, the atlas.
Then we saw protein quality control failing, leading to amyloid buildup and inflammation, this amyloid immunoglobulin complement axis.
The breakdown of proteostasis.
We found universal protein markers of aging, allowing for accurate proteomic clocks.
The UDPAs and UPAs.
And this striking inflection point around age 50, suggesting a real biological transition.
Yeah, that midlife shift.
And finally, the identification of the aorta as a potential early aging xenohub, driving systemic decline, partly through circulating factors like GAS6.
You've got it. That's the core story.
So the really exciting part of this, thinking forward, is if these proteins like GA6 are actively driving aspects of aging, could we target them?
That's the million-dollar question, isn't it? And the answer is potentially yes. If aging is being spread by these specific molecules,
then we don't necessarily need incredibly complex gene therapies. We could go after the proteins themselves.
it opens up that possibility. You could imagine developing, say, neutralizing antibodies that bind to GA6 or these other senoproteins in the blood and just take them out of circulation, stop the signal.
Or what about targeting the source, the senescent cells in the aorta that might be producing these factors?
That's another exciting avenue. People are already working on senolytics drugs to clear out senescent cells, but this research also hints at maybe senolytic vaccines.
Vaccines against aging cells, how would that work?
Well, if senescent cells have unique proteins on their surface, maybe some of these UUPAs we talked about, these xenoantigens, you could potentially train the immune system to recognize and eliminate those specific cells.
Wow. Using the body's own defenses to clear out the troublemakers. Targeting aging at the protein level, either the messengers or the source cells.
It's a very different way of thinking about anti-aging therapies, much more targeted than just general antioxidants or something.
That is a really powerful thought to end on. The potential for truly specific mechanism-based interventions based on understanding the proteome of aging. Thank you. This was incredibly insightful.
My pleasure. It's fascinating work.
And thank you, our learner, for joining us for this deep dive. Please remember to like, subscribe, and share your support. Really helps the SANS Science experience. We'll be back soon exploring more cutting-edge science.